All right, everybody. So in the previous video, we went over the innate immune response, also known as the non-specific immune response. In this video, we're going to go through the adaptive immune response, also known as the specific immune response. And it's specific because the cells involved are looking for very specific antigens on the surface of the pathogen or the infected cell or the cell that is a tumor. Will also have very specific antigens that these cells are trying to target. Now, like I said, it's very specific to certain types of antigens, but that doesn't mean it acts alone. The adaptive immune system can also initiate the innate immune system if it needs to. All right, it could have a role in inflammation. All right, so if one of these adaptive cells finds one of its targets, then it can release different chemicals to initiate inflammation response or activate some of the complements that are nearby. Now, this is a very powerful immune or part of the immune system. All right, your adaptive immune system can really do a good job killing a lot of the foreign bodies that are in your body. But the problem with it is that it needs to be primed in order for it to work. So usually the first time you're infected with something, this can't do much. It needs to actually detect that antigen and then start making this adaptive response. But once that happens, then your body's going to recognize that pathogen again in the future, which means you're going to get rid of that pathogen much more quickly the second time around than you would with the initial infection. All right, so again, we're looking for very specific antigens with our cells that are involved with this. But one of the best parts about this is that once the cells that have that very specific antigen detect that foreign body, then they're going to make tons of themselves, which are going to last throughout your life. So then you're going to have tons of cells that are all primed for that one specific type of antigen. So if you're ever reinfected again, you're going to be able to attack it. And this is systemic. This happens all over your body, unlike the innate immune response, which usually happens at very localized areas in your body, wherever the infection is occurring. And we say it has a memory because it's able to do this. It will remember that pathogen again in the future, so it can attack it immediately the second time you're infected with it. Now, there's two major branches to the adaptive immune system, and it has to do with the two cells that are involved. So we have humoral immunity, which is going to be controlled by your B cells. And humoral immunity is talking about the antibodies that are going to be produced by your B cells. Humoral just refers to solutes, and the solute we're talking about here is those antibodies. So we're releasing a bunch of solutes that are antibodies into our plasma, which are going to float around our system and attack this foreign body. Versus cellular immunity, which is, or cell mediated immunity, this is where your T cells come in, which go and directly attack whatever they're looking for. Whatever that specific antigen is, it will attack the cell that it's on, whether it be the foreign pathogen itself or an infected cell or a tumor cell. All right, but collectively, this is known as the immune response, which might seem weird that we call the adaptive immune response the immune response, but it's because the adaptive immune response was found first in science. We discovered the adaptive immune system way before we discovered the innate immune system. It wasn't until later that we realized there was other parts of the immune system. And we just thought all immunity was this kind of very specific sequence of events where you have this initial infection, your body kind of primes itself for that infection, and then it can fight it off later. So don't let the term confuse you. Whenever we say an immune response, we're talking about the adaptive immune system, while your innate immune system has different, or also is part of your immunity. Okay, so all of this is because of the receptors that are on your B cells and T cells. They're very specific to a certain type of antigen. And if those receptors attach to that antigen, then you're going to have your body start to prime itself to kill that antigen or whatever cell that antigen is a part of. All right, so we have names for the receptors that are being used by both your B cells and T cells to attach to these antigens. We call them B cell receptors or T cell receptors. So BCR versus TCR. So you can see those abbreviations a lot. And these are going to go and look for that antigen. Now, there's a difference between B cells and T cells, though. B cells can make direct contact with that antigen and then start priming. That's okay. T cells can't do that. T cells must have the antigen presented to them by another cell. So if it's just an antigen floating around, B cells can detect it and start doing something. T cells need to have that antigen presented to them by another cell, and that is going to help with their priming. Now, the reason for this is because T cells have another type of receptor on them, not just their TCRs, but next to the TCRs, we're going to have molecules called CD molecules. And these CD molecules look for another type of antigen or another type of receptor on other cells which is going to be required for your T cell to recognize the antigen that it's going to detect on its TCR. This might seem a little complicated at first, but once you start seeing images of this and we go through this a little bit more, that will make much more sense. All right, but before we do all that, let's just kind of have a better understanding of what antigens are. Antigens are simply just molecules that act as flags on the surfaces of your cells. All right, so there's two major types of antigens. We have self-antigens and we have foreign antigens. Self-antigens are just a bunch of different molecules that your body makes naturally that are also going to be on the surface of your cell that other cells can detect and be like, hey, you're with us. All right, you're part of ourself. So we're not going to attack you. Versus foreign antigens, which is something your body won't produce. It's going to be produced by a foreign body, and that is going to stimulate some sort of response with your immune system. So another terms for this is for foreign antigen, we also call it non-self antigens because, again, it's not yourself that's creating them. Okay, so all cells present antigens on the surface of their cells. Every nucleated cell in your body is going to make antigens. All right, and put it on the surface of the cell. And if it's an antigen that your body knows and that your body makes naturally, that's going to be a self antigen. And if it's an antigen that your body doesn't recognize and came from a cell that's not you, then it's not going to be non-self or foreign antigen. Now, all nucleated cells do this, but we also have a subgroup of cells called antigen presenting cells or APCs that do it a little bit more refined and more specifically. And this is going to help the adaptive immune system really initiate a response. So the major cells that can do this more specifically or more specialized in presenting antigens are your dendritic cells, your macrophages, and your B lymphocytes. So even your B lymphocytes can stimulate the priming of your T cells. Now, these antigens are not just floating around in the surface of your cell, they're actually held there by another type of protein called your major histocompatibility complex. So it is a protein that all cells make. So we initialize as MHC. It's a protein that all cells make and they make tons of, which are going to collect antigens and bring it to the surface of the cell and hold that antigen up outside the cell for other cells to come by and detect. All right, so there's two major classes of MHCs. We have class 1, which all cells can do. Any nucleated cell can make that type of complex, versus MH2s, which only your antigen presenting cells can make. All right, so again, this is majorly why we have two different categories, because all cells make MH1 or MHC1. Only your antigen presenting cells like your dendrites, macrophages, they're going to make MHC2. Okay, so how this all works. So we call this MHC docking, formation and docking. So MHCs are proteins that are going to be made by your rough endoplasmic reticulum, where all proteins are being made, right? It's made in your endoplasmic reticulum. This is a naturally occurring protein that your cells make. All right, so it's produced in the RER. And as this is happening, you also have parts of different proteins that have already been made that are not complete or broken apart in your cell, because your cell is constantly breaking down proteins it makes once they're not used anymore, or if they just are not functioning. And so little pieces of peptides fall off of those proteins. And those peptides can then bind with your MHC. So let's say this is a normal cell. This would be your MHC1 class. So peptides bind with the MHC1. That MHC1 and peptide complex is then going to be packaged by your Golgi apparatus. And with that Golgi apparatus, that little vesicle that is released is going to go to the surface of the cell where the vesicle kind of just fuses with the plasma membrane and presents this MHC complex on the surface of your cell. So you have your MHC molecule that is in the plasma membrane holding up the antigen that is now your self antigen because it came from one of the proteins that your cell naturally makes anyways on the surface. So now other cells can come around and look at this flag, this antigen, and be like, oh, that's something we already make. That's part of us. All right, so that's a self antigen. So we have millions of different types of antigens that could occur on the surface of the cell because all it is different pieces of peptide from broken down proteins that we've created. Now let's see what happens in an unhealthy cell if there's a cell that's infected. So your cells that are infected want to broadcast, hey, I'm infected, to your immune system, so your immune system come and take care of that cell because the cell is most likely going to be dead anyways. So the example we're going to use here is a viral infection. So what happens is a viral particle comes in, all right, it's going to enter the cell and try to infect the cell. Again, you're still going to be making those MHC class 1 molecules in your rough endoplasmic reticulum, just like we did before. However, as the virus starts to take over your cell and replicate itself, it's going to try to make proteins of the virus's DNA and RNA that is going to help the virus out. Just like before, those proteins can be broken down by normal cellular processes, like a proteasome or proteasome, which is just designed to break down proteins. Those proteins that are released that came from the virus's genome or transcriptome is going to then be broken down into peptides. Those peptides will then bind with the MHC class 1 molecule, and the same process occurs. So we have this peptide MHC complex that is packaged in the Golgi apparatus, which is sent to the surface. But now, instead of having peptides as an antigen that came from us, it's a peptide that came from a virus or a bacteria or anything else that infected inside the cell. So now if another cell comes along and tries to look at this flag, this antigen, and says, hey, we didn't make this type of peptide, this is not from us, they know that this cell is infected and something can be done about it. All right, so this, again, all cells do this MHC1 class complex. So again, what we saw before was what's happening normally when it's not infected. You have self-antigens coming to the surface from the proteins your body usually makes. With the unhealthy cell, we have now proteins being antigens that came from a viral infection or some other source besides our own gene or DNA. Then we have class 2 MHC. So MHC class 2 is, again, from those antigen presenting cells, whether they be dendritic cells, macrophages, or B lymphocytes. And these are going to do this in order to present that antigen to one of the adaptive immune cells, whether it be other B cells or whether it be the T cells that can then go and hunt that antigen. So very similar process like we saw before, but it involves phagocytosis here. So if you remember with phagocytosis, something that is ingested is going to come in as a phagosome, in that vesicle called a phagosome, that binds with the lysosome to create a phagolysosome. And then that phagolysosome will then disintegrate or digest that bacteria or virus that's caught inside that phagosome, or now phagolysosome. When it breaks down, things that are released are like different types of peptides that came from that bacteria or virus. And so now you have peptides floating around again. Here, we have a different class of MHCs that have slightly different conformations that can easily bind with anything from a lysosome, or excuse me, bind from anything from a phagolysosome. So instead of just the MHC2s binding to a peptide, then going to Golgi apparatus and then going to the surface, instead we make our MHC2s, it gets packaged in our Golgi apparatus, and then that is going to be included in our lysosome. So we have these MHC class 2s in the lysosome already. So when the phagolysosome forms, the enzymes of the lysosome break down the bacteria, and then that MHC2 is going to bind to any peptides that came from that degradation. And then from there, it can go to the surface and display that antigen on the surface of the cell. All right, so now we're going to talk about different types of lymphocytes. So if you remember different types of T lymphocytes. So with T lymphocytes, these are the ones that are going to either try to directly kill a cell, or it's going to kind of help mediate the rest of this adaptive immune system or adaptive immune response. Now, there's two major types of T cells. We have helper T lymphocytes and cytotoxic T lymphocytes. The cytotoxics are also known as killer T cells, which you've probably heard of before. But the major difference between these two cells is the CD that they make. So it's something that we mentioned a couple slides before. Not only do they make their TCR, right, their T cell receptors that are going around looking for a very specific antigen, they also make something called a CD protein that is on the surface very close to that TCR, that is going to help make sure they're binding to another cell and not just the antigen itself, which is actually really important to make sure you don't accidentally start killing your own cells. So what's happening here is, again, these TCRs are very specific to a very specific antigen. If it finds that antigen, it will not hold on to that bond unless this CD protein attaches to either MHC1 or MHC2. All right, so the major difference between helper T cells and cytotoxic T cells from a structural standpoint is which CDs they have. So helper T cells have CD4 in their plasma membranes, which directly looks for the MHC2 complex, while cytotoxic T cells have CD8, which directly looks for the MCH1 complex. All right, so if you remember, MCH2 only comes from those antigen presenting cells, those macrophages, those dendritic cells, or the B lymphocytes. So helper T cells are only going to be primed or activated if they come in contact with one, that antigen they know how to bind to, and two, that antigen was presented by one of those ACPs, or say APCs. Okay. All right. Well, the cytotoxic T cell can bind to any cell in the body that produces MCH, which is all nucleated cells can do this. All right. So if it has an MCH1 and it has a foreign or that specific antigen it looks for on that MCH1, then these will be initiated. Now, the response is a little different between these two cells. The reason helper T cells are called helper T cells because they're there to kind of mediate both the cellular and the humoral immune response for your adaptive immune system. And not only that, they can also help with the innate immune system, kind of attract more blood or white blood cells to the area to have an innate response as well as this adaptive response. While cytotoxic T cells are there to go kill whatever cell that they attach to. Or if they find that antigen that they're very specific to and bind to it, they're going to try to kill that cell, and they do it very similar to how a natural killer cell does. They can release perforin, they can release granozymes. So all those things would happen, creating apoptosis, which is a very controlled way of killing a cell to make sure that pieces of the cell don't go flying everywhere and you have further infection. All right, so just to recap this real quick, we have these TCRs, these T cell receptors that are specific to a certain type of antigen. Now, these antigens need to be presented on another cell. And the reason for that is because whether it's CD4 or CD8, those two proteins need to bind to MHC1 or MHC2, depending if it's 4 or 8. CD4 binds to MCH2, while CD8 binds to MCH1. When they bind, then the connection between the T cell receptor and the antigen can go ahead and complete. When this happens, then you're going to have stimulation of these cells, which are going to then prime the cells for adaptive immune response. Next, we're going to talk about T cell formation and selection, because you don't want T cells that are out there that are going to attack your own cells. That would be bad. But you also want to make sure you only have T cells out there that can attack to some sort of specific antigen. All right, so again, if you remember, T cells are made in bone marrow along with B cells. However, B cells mature in the bone marrow, while T cells migrate to the thymus where they mature. So the T cells that are released from the bone are called immature T cells because they're not fully developed yet. When they get to the thymus, that's when development is going to happen. Now, I found this image in an old textbook. I could not find a good image of this online, so I'm sorry for the photocopy here, but that's the best I could do. But this is a really good image kind of breaking down this whole formation and selection process. So what we're doing when we bring our T cells to the thymus, we want to develop them so they have a specific receptor, and they have either CD4 or CD8, so they become helpers or cytotoxic T cells. And we want to make sure that they don't attack our cells. When this happens, we consider these cells to be immunocompetent. They're competent immune cells. So we have this whole selection process that occurs to make sure that these cells are able to do what they're supposed to do. Now, I get this a lot with students. They always ask me, how does your T cells learn the antigen that attacks? Like whenever you're infected with the foreign body, how does your T cells learn to attack that one antigen? And the truth is they don't. That is not how this works at all, actually. So when your cells are being developed in the bone marrow, they go through this thing called gene recombination, which is a random switching of their genes. They'll switch the order of their genes in their genome. And when they do that, they're going to have a very random created T cell receptor. So each T cell that cell that makes a T cell receptor makes a completely random receptor that can bind to something random. All right, so it is kind of like a crapshoot. You're just making a bunch of different types of receptors, and hopefully they're not going to attack the own body, and that they will attack something else. All right, so random process of just random reorganization of the gene that makes up the TCR. TRC. No, TCR. I had it right the first time. All right, so that is gene recombination. Random TCRs are created. When that happens, then those cells are released to the thymus, and this is where you are going to have a selection process occur. The cells are going to fully mature, and then you're going to make sure that the cells don't attack, that random TCR does not attack your own cells or self-antigens, but are also capable of binding to those MCH or MHC complexes. All right, so that's a two-part process there. So these immature cells have a random TCR, and when they are released, they actually have both CD4 and CD8. All right, so they're not yet defined to be a helper or cytokinetic or cytotoxic T cell yet. All right, so once we're in the thymus, this is when the selection process occurs, and it's a two-part process. We have positive selection and negative selection. So for positive selection, this is really the first stage of all this. Inside the cortex of the thymus, if you remember that from the lymphatic system, you have a bunch of epithelial cells that are creating that cortex. Those cells make MHC1 and MHC2 proteins. All right. So when that happens, what is going to happen with the T cell that comes through there? It's going to make sure that that CD4 and CD8 can bind to those MHC proteins that are in those epithelial cells. So once they bind, they're considered competent for MHC or for MHC. So if they're competent for MHC, the ones that don't bind get destroyed. They're killed through apoptosis. All right, so we have just weeded out everything that won't be able to bind to MHC. Once we're done with the positive selection, we have killed all the cells that are unable to bind to MCH, whether it's one or two. Then we go to this negative selection that happens in the medulla of the thymus. In the medulla, we have a bunch of dendritic cells, and those dendritic cells are going to produce a bunch of self-antigens on their surface as antigen presentation that we talked about a few times now. So you have a bunch of self-antigens on the surface of these dendritic cells. So now if a T cell binds to that dendritic cell, we know it's a bad T cell. All right, you don't want your T cells to attack your own cells. So we do it in this controlled fashion with the dendritic cells, where if that occurs, then apoptosis can happen to that T cell. So this is called negative selection because the T cells that make it through here without binding are fine. They're not going to be killed because they're not binding to a self-antigen. But the ones that do bind need to be destroyed because if they are released into the system, they're going to have an autoimmune response. You're going to have a self-attack, or it's going to attack your own cells, which we're trying to do away with. All right, so the cells that bind here are destroyed, while the cells that don't bind can pass through. Once that happens, the cells will then either lose their CD4 or CD8 to differentiate to be a helper or cytotoxic T cell. These cells will then migrate. They're called mature cells, but we still consider them naive cells because they've never encountered the antigen that they have made a random assortment to bind to. All right, so we consider these naive cells because that antigen has not been presented yet to this cell. It's just a random TCR that may or may not bind to an antigen, some specific antigen. So these naive cells that are now immunocompetent and now mature are carried to secondary lymphatic organs, like a lymph node, like down here. And that's where they're stored. So usually when you have an adaptive immune response, it's happening in these secondary organs because something has brought that antigen to these areas and found the correct T cell to do something about it. All right, so now we just talked about how we could select for immune competent T cells. All right, so we talked about how we looked for T cells that can bind to MHC complexes. That's important because you need that in order to bind to a antigen presenting cell. And we talked about how we make sure that they don't bind to one of your self antigens because that's bad, because then those T cells are going to go around killing your own cells. We don't want that. So we selected for those through positive and negative selection in your thymus. Once we do that, then those T cells will differentiate into be either helper T cells or cytotoxic, depending if they have CD4 or CD8 proteins on their surface. Those are then transported to secondary lymphatic organs where they're stored. So this is where they hang out mostly, which is waiting to be initiated or waiting to be activated by that very specific antigen that random TCR can attach to. All right, so we're looking at T cell activation here, and it's really a two-step process. It takes two signals to activate a T cell and make it have an adaptive immune response. So, and they're kind of related, whether it's a helper or a cytotoxic T cell. You can't have one without the other. So with the helper T cells, let's start there. The first signal for a helper T cell is from a non-self antigen that it can bind to. So that very specific TCR can bind to this very specific antigen that's not from your own body. Now, these antigens are presented to it by those antigen presenting cells that create the MHC2 complex. So we talked about how MHC2 can take a foreign peptide, add it to the MHC2 complex, and present it at the surface of these cells. Well, these macrophages, especially the dendritic cells, do this really well. they will go and try to find the T cell that can bind to that antigen. They go around, T cells are matching up with dendritic cells, and if they can bind, then, or if they can't bind, they just bounce off each other and they keep on going looking for the next T cell over and over and over again until finally one of these T cells or the helper T cells bind to that antigen with their very specific receptor. So that's signal one. You need that binding between the antigen and the receptor before you go to step two. For helper T cells, signal two is actually really easy. There's just other receptors that are on the cell that can bind with that ACP cell. All right, so if we have an antigen bind with the receptor and we're able to bind to other receptors that are on this APC, we know for a fact that this antigen is being presented to us by one of these APC cells. Okay, so it's an important second signal. That way, it's not just an antigen that's attaching and then you have this response, because you don't want that. You're going to have things go out of control if that happens. So you need to make sure your antigen is presented by an APC. Having the second signal that detects other receptors on an APC cell is a great way to do that. Okay, so what happens after we activate a helper T cell? Well, they're going to start proliferating. They're going to clone themselves. You're going to have tons and tons and tons of helper T cells. Again, this is one of the reasons why our lymph nodes get so swollen when we're sick, because this is happening. You're making tons of these cells through division. And these new cells that are being formed are exact copies of that original cell. And so they have that exact same TCR that attaches to that very specific antigen. So you're going to have two different cells come from this. You're going to have active helper T cells, which are going to be released, and they release something called cytokines that are going to help with the immune system or help with the immune response. Or you're going to have memory T cells that just stay in your lymph nodes forever until they die. And they're going to be there waiting, seeing if this antigen ever presents itself again. All right, so we went from this one cell that is very specific, from tons of random cells that are all very specific, to proliferating this one cell to have tons of it. So now your body's really ready for that antigen or whatever that pathogen is. All right, so that's helper T cells. Cytotoxic T cells, the one that actually does the killing, is very similar. So signal one is, again, being presented that very specific antigen that it can bind to. All right, it's a non-self antigen that is very specific to its TCR. But here, we're not looking for a TCR that's on an MHC2 complex. We're looking for an antibody or antigen that is on an MHC1 complex. So any cell can stimulate a cytotoxic T cell. Or sorry, any cell can give the first signal to this activation. It doesn't take more than that to actually activate this T cell. The second signal is coming from those cytokines that were released by that original helper T cell. So this is the only way cytotoxic T cells go rampant and start killing everything that can find an antibody to. All right, so these cytotoxic T cells need to first bind to that specific antigen for first signal, but also receive cytokines from a helper T cell that already has been activated. So you need this kind of checks and balances system to make sure your cytotoxic T cells don't just start activating randomly. All right. So again, just breaking this down, with the helper T cells, the first activation is from the binding of the antigen and the TCR. The second activation is from other receptors that are on the APC binding to that helper T cell. This allows that helper T cell to know, one, this antigen exists that I know how to kill, or I know how to find, I should say. And this antigen was presented to me by one of these dendritic cells or one of the other APCs. So I know my body has already tried to attack this foreign body or this foreign pathogen, and now they need my help, and I have the tools to do it. And so once that happened, helper T cells then release cytokines, which is a suite of different proteins and molecules that are just released out into the blood or in the tissue. Now, when it comes to cytotoxic T cells, similar process. You're going to have an antigen bind to a very specific, or I'm sorry, you're going to have a TCR bind to a very specific antigen. This is only going to bind with MHC1 because of the CD that it has. And that's signal one. And then signal two is the cytokines that are being released by your helper T cell. So a helper T cell needs to be activated in order to activate a cytotoxic T cell. So now the cytotoxic T cell knows, one, I found the antigen that is being presented to me on a cell. Two, I found the cytokines that a helper T cell had detected. So this helper T cell went through the same checks and balance process. So I know I need to activate now and go kill whatever this is. And so just like the helper T cells, these cytotoxic T cells are going to proliferate. They're going to divide over and over again. Again, this is usually happening in those secondary organs of the lymphatic system. So what happens is that you're going to have some of these clones be activated and go and hunt down that antigen, while other of these cells are going to be memory cells and stored in those secondary organs again, waiting to see if that antigen ever presents itself again. So again, we went from all these random T cells that have random receptors on it. Each individual one has a different type of receptor that's completely random, to finding one that matches this antigen that we know is from a pathogen. And once that happens, then we can have tons of that very specific cell in your body that can go around hunting for that antigen. All right, B cell activation is pretty similar. All right, again, B cells are the ones going to make antibodies. They're not going to go hunting the cells themselves or hunting the pathogen themselves. They're going to make antibodies that kind of do all the work. So B cell go through the same very similar selective process as the T cells, except B cell selective process doesn't happen at the thymus. It actually happens in the bone marrow, hence B for bone marrow. All right, that's where the selection occurs, is in the bone marrow. But let's talk about what signals their activation. Again, it's a two-step process. Signal one is when the antigen is going to bind to that very specific receptor on the B cell. All right, so you're going to have antigen and receptor bind, and in for B cells, we call them B cell receptors, BCRs, instead of TCRs. And when this happens, that B cell is going to engulf that antigen, because the antigen does not need to be a part of a cell here, unlike a T cell. The antigens do not need to be presented to a B cell like a T cell does. So you could just have the antigens floating around your body, which may have came from, that phagocytosis of macrophage or a neutrophil, and that exocytosis of debris. Part of that could have been one of these antigens that match up with the receptor that is on this B cell. And when that happens, that B cell receptor will attach to that antigen. It will then engulf that antigen, and then it will start presenting that antigen on its surface, just like other APCs would. All right, when this happens, when you present this antigen now, it is presenting this antigen on surface. Again, same thing with MHCs. It's going to bind to one of these helper T cells. So when this happens, when this B cell presents this antigen and binds to one of the helper T cells, again, that helper T cell is going to release those chemicals, those cytokines. Those cytokines are then going to stimulate this B cell. So this is signal two. Signal one was presenting this antigen. Signal two is when you attach this antigen to a helper T cell, which releases cytokines, which stimulates this B cell. If this B cell gets both those signals, gets an antigen it can detect, and it gets cytokines from giving that antigen to a helper T cell, then it's going to start proliferating, just like we saw already. All right, you're going to have active B cell clones, and then you're going to have memory B cell clones. And same thing, those memory B cell clones are going to hang out inside your secondary organs, waiting for other times where this antigen is presented. Not only that, those active B cells also stay in a lot of these secondary organs. They don't go traveling, and nor do they need to, because they're not there to go hunt down a cell. Instead, they're going to make antibodies that are released into the blood or released into your lymph and go around trying to, and those antibodies are going to go and try to find that foreign pathogen. Okay, so once this happens, once we have active B cells and memory B cells, now we have tons of B cells with the same very specific receptor that can make a bunch of antibodies of that very specific receptor and look for that very specific antigen. So now we have antibodies being produced that can go find that specific antigen. So we have a name for those cells that can produce that antibody. So we call them plasma cells. So simply put, an activated B cell that is making antibodies is now known as a plasma cell, because it's releasing these antibodies into the plasma. All right, plasma cells. And these antibodies are the ones that are going to go around looking for that very specific antigen on that foreign pathogen. All right, so a little bit about antibodies. So antibodies are immunoglobulins. All right, we saw a whole suite of immunoglobulins, but this is just a very specific type of immunoglobulins that are going to go around and attach the antigens on other cells for the purpose of destroying that antigen. They are Y-shaped, just like we talked about before. All right, so they do make a Y. All right, and we have two different regions of this antibody. So we have something called the variable region, which is usually one of these. We have two of them. They're both of these arms on the Y of this antibody. And the variable region is what's going to change from B cell to B cell to B cell. It's all going to have very specific binding sites for very specific antigens. So depending on what B cell made which antibody, this is going to change, and it's going to bind to the very specific antibodies on different cells. And again, making sure that antibody does not attack that self antigen. All right, these antibodies are only going to be made that can attack non-self antigens. So those are variable regions. Then we also have something called a constant region, which is pretty similar from each antibody. All right, there's only a handful of different types of constant regions that your antibodies can produce. And these are going to have different effects on the body and ultimately kind of control how your antibody is going to deal with that antigen, or even so, that pathogen that has that antigen. All right, so variable region is the region that's going to attach to a very specific antigen, and it's determined by the very specific B cell that it came from. Constant region is going to determine how that antibody is going to actually affect that antigen. All right, so we have three classes of these antibodies. We have neutralization, agglutination, and precipitation. And it's going to vary depending on what we're attacking, really. So neutralization is part of this whole antibody's power, all right, its ability to stop these pathogens that it knows how to attach to. Because you can make so many of these antibodies, once you release them into your bloodstream, you have enough to coat the entire pathogen. So here in this example, they're using a virus, and it has these antigens on the virus. All these antibodies know how to attach to that antigen, and then you just overwhelm the virus with this. When you have a completely coated virus, that virus cannot infect another cell because it can't even detect the receptor of your host cell to even get in or impregnate that cell. So you're kind of blocking the virus's ability to enter a cell with neutralization. You neutralize the virus. Comparing that to agglutination, which we've already talked about, is when these antibodies are able to bind to multiple different pathogens that has the same antigen. So we see this a lot, especially with bacteria, that if you have a bacteria that has a specific antigen and that bacteria started replicating, making tons of itself, well, you can kind of neutralize that or stop that bacteria by having these antibodies bind one and then cross-link it to another, which can cross-link it to another and cross-link it to another. And eventually you have all of these different bacteria that are just clumped together into one mass, or agglutinate into one mass, which can then sink out a solution. And once that happens, usually other immune cells come in and take care of that, all right, then go around and killing those bacteria. And then finally, something very similar to agglutination, we could have precipitation. It's the same idea as agglutination, except for here, we're trying to get rid of some of the particles that are left behind by these foreign pathogens. This is especially true when it comes to viruses, because you could have just a little tiny DNA of the virus still be active, and you might still have a problem. So one of the things the antibodies will do, the antibodies will do, if they find just the antigen itself that it's specific to, it doesn't have to be a cell in this case, so it's not a cell in this case when it's precipitation, it's just some solubilized component that is floating around the blood. They can start collecting these soluble particles, and then once enough of them are collected together, they drop out or precipitate out of solution and fall out of your blood. And again, some other cell can come around and then take care of it, either through usually through phagocytosis. All right, so those are the three major ways that your antibodies can affect or impact the foreign substance. So again, they can neutralize it by preventing its ability to interact with one of your cells. They could agglutinize it by pulling the cells out of solution and then having something else take care of it later on. Or they could precipitate it, same idea, except not cells, just solubilized particles that they're trying to get out of solution. All right, then ultimately what happens is going to be determined by that constant region of the antibody. All right, so we have three classes of what these antibodies can do based on that constant region. So we have complement fixation, opsonization, which is a fancy word, and activation of natural killer cells. All right, so complement fixation is pretty straightforward. So we have those active or variable regions that are very specific to some antigen, so they bond to that antigen. And when that happens, the constant region, if it's a constant region that is known to bond with complement, a complement protein can bind to it and initiate that complement enzyme cascade that we've talked about previously. So here, those antibodies are kind of stimulating your innate immune system. All right, that was that classical pathway that we talked about previously. Another thing that antibodies can do, the constant region could help with opsonization, where again, you have this constant region sticking out that phagocytic cells can know how to bind to. And when they do, they can really grab onto that foreign body or foreign pathogen and then engulf it. All right, so it's kind of like giving them something extra to hold on to, so it's easy to catch these little things that are crawling around and floating around your system. All right, so that's opsonization. All right, and then finally, what one of these constant regions can do is stimulate a nearby natural killer cell. All right, so again, we have this binding between the active or the variable region that is specific to some sort of antigen, which is only going to be specific to antigens that it knows. And since selection happened, we know it's from a foreign body. And this constant region can then stimulate any nearby killer cells or natural killer cells that come in contact with it. And when this happens, it tells the natural killer cell, hey, you need to kill what I'm attached to. And natural killer cells will then release those enzymes, whether it's perforin or granzymes, that are going to cause that cell to go through apoptosis. And again, really controlled method of killing the cell to make sure anything inside the cell doesn't just go flying everywhere. All right, so that's it for adaptive immune system. So hopefully having these videos will help you a lot with going back and seeing some of the stuff again, because I know it's a lot. It can be kind of complex, but it's very fascinating how your body is able to ward off foreign pathogens. So hopefully having a video you can go back and like see these things a couple of times. It's really going to help you understand these processes. So you really need to be able to understand what cells are doing, whether it's for the innate and both the adaptive immune system. You should have a good idea on how your body presents antigens to begin with, and the difference between self and non-self antigens, and how your different immune cells detect that and what they do. Know the parts of inflammation, that's also important, because you need to get all these white blood cells to these areas, especially, and a lot of those antimicrobial proteins to these areas too. Understand other parts of the innate immune system, like inflammation or fever, or understand why they're so important. And definitely understand how we're able to go from a T cell or B cell that has this kind of random generation of a receptor, T cell receptor or B cell receptor, and how we're able to make sure we select for only T cells and B cells, well, more specifically T cells, how we select for T cells that we make sure that they can bind to an antigen presenting cell, and to make sure they don't bind to your self antigens before they're released into the body. And then understand how activation works too. What needs to happen in order to activate one of these T cells and B cells? What are the two signals? What cells are involved? And eventually, what is the outcome? All right, what is an active helper T cell or active cytotoxic T cell, or a memory helper T cell or a memory cytotoxic T cell? And the same thing with the B cells too. And what is a plasma cell? All right, and also understand how antibodies help with all of this. Antibodies are major players for your immune system. All right, they are constantly being released. If your body knows it's come in contact with some sort of foreign pathogen again, it will release antibodies in your system. And if those antibodies ever come in contact with one of these foreign pathogens again, it could be immediately responding to that. It will immediately attach that thing as it enters, and then your body can take care of it in many different ways, like we discussed. All right, well, hopefully you found this interesting. I find this fascinating, how your body is so adept to taking on these foreign pathogens that enter into your system. If something gets past one of those primary barriers, those first line defense, your body has so many things and so many mechanisms involved to take care of that foreign pathogen. And whether you get sick or not is really a balance of your immune system versus how infected you are, right? How much of a virus or how much of a bacteria gets into your system, and does it overwhelm these natural processes you have in your body to kind of combat that? And if so, how long does it take before your immune system starts to overtake those bacteria or viruses? All right, so hope you enjoyed this. Again, I found this very fascinating, so hopefully you did too. And I will see everybody next week. All right, take care.